Hello, my name is Arturo Sala. I'm a professor of translational cancer biology at Brunel University London, and I'm a recipient of an Oracle Cancer Trust Grant Award. One of the research interests in my laboratory is adenoid cystic carcinoma, a form of head and neck cancer that mainly affects salivary glands, but can also originate in breast glands. The cancer is slow-growing, but relentless. It is not stopped by standard chemotherapeutic drugs and has tendency to metastasize in distant organs or to grow along cranial nerves, leading to patient death. One of the molecular characteristics of adenoid cystic carcinoma, or ACC for short, is the activation of an oncogene called MIB. Virtually all ACC tumors present activation of MIB, which is required for the growth and survival of ACC cancer cells. Unfortunately, there are no drugs usable in the clinic that can block MIB. Furthermore, there are few cell systems to study ACC. So, in the project funded by the Oracle Cancer Trust that we have just completed, we try to address these points. Firstly, we have generated a new cell system based on breast cells containing an inducible MIB oncogene. Using molecular profiling, we have ascertained that the cell system is comparable to genuine ACC tumor cells when the MIB oncogene is activated. This new cellular model of ACC is easier to manipulate and more cost-effective than previous systems. Therefore, it will be useful to the scientific community interested in studying ACC. A second aim of the project was to investigate the relationship between MIB and downstream target genes that could be used as potential therapeutic targets. Using the cell model generated in our laboratory, we have identified the gene encoding the protein kinase BAB1 as a MIB target. Kinases are enzymes that add phosphate groups to other proteins or molecules. This is important because it is relatively easy to find small molecule inhibitors of kinases. We have therefore used a small molecule inhibitor of BAB1 and found that this can strongly inhibit the growth and induce death of patient-derived cells and organoids in a MIB-dependent manner. In the future, we plan to validate these results in mouse models of the disease to assess whether the BAB1 inhibitor can be used to sensitize ACC tumor cells to standard chemotherapeutic drugs or radiotherapy, increasing their efficacy. Completion of these studies could lead to clinical trials in which the BAB1 inhibitor will be added to current therapeutic strategies, hoping to improve patients' long-term survival and quality of life.